Yeah, it's my great pleasure to welcome you all again to the Club EvMed. And again, just as a reminder, the goal of Club EvMed is to connect the evolutionary medicine community and share the exciting research that is done by all our colleagues. And the idea is to have these Club EvMeds as informal as possible. So you're welcome to pose your questions either at the end, but you're also welcome to actually pose questions today in between. So both speakers are happy with being interrupted from time to time, if you don't overdo it. Another reminder, so the Club EvMed is organized by the International Society for Evolutionary Medicine and Public Health. If you're not part of the society, please join us. The society also organizes very exciting meetings this year in the UK. And it is also responsible for the journal Evolution, Medicine and Public Health, which also offers a very fantastic opportunity to publish your papers on this topic. The Club EvMed is additionally supported by a variety of other evolutionary medicine centers, including the center that we have at the University of Kiel in Germany, and that's where I'm from. So just as a last point, so please participate. The idea is that we have a discussion after and during the talks, the presentations, and you can join the discussion either by writing your question into the chat. I will then call upon you so that you can then speak up and present the question, or you raise your hand, which we hopefully notice also during the talk. But these are the two options: raise your hand or write in a question into the chat. Then it's my great pleasure to introduce the two speakers of today. One of the speakers is Sada Boyd-Forsyth, currently at Emory University. Sada did her studies in North Carolina and then continued for a PhD at North Carolina Agricultural and Technical State University. And she thereafter did her first postdoc in the lab of Pamela, our other speaker today. And if I understood correctly, then relatively recently, Sada, you moved to Emory University, joined the lab of Graham Conn, where you're now studying aminoglycoside resistance in Pseudomonas aeruginosa. Pamela Ye studied at Harvard, then crossed the United States to do her PhD at the University of California in San Diego. Then she moved back to the other side of the US to do a postdoc at Harvard University. And then she didn't get bored by always crossing the entire country. So she moved back to the West Coast, and she is a group leader at UCLA in Los Angeles since 2013. And it is particularly fascinating that Pamela is not only studying evolutionary medicine topics, like what she will present today, the evolution of drug resistance in urban and agricultural areas, but Pamela is also looking at the effect of human activities on the evolution of birds in urban environments. So unfortunately, we cannot hear about the latter, but we're very much looking forward to hear from both of you about antibiotic resistance evolution. And the floor is yours.
Thank you so much, Heinrich, for that really kind introduction. And also thank you, Heinrich, for the invitation to speak here. And thank you, Johnny, as well, for organizing this fantastic Club EvMed. I'm thrilled to be here and grateful for the invitation. And also want to say I will talk for about 5 minutes, but most of the time will be Sada Boyd who will be talking. She's a fantastic early career researcher, has been a postdoc for a few years. She'll be on the job market. So if any of you guys in your departments are looking, there's a phenomenal opportunity to get just a great scientist, if I can make that quick plug. Okay, I will be talking, as Heinrich said, about evolution of antibiotic resistance. And I want to focus, excuse me, I'm just getting over this like extremely long cold. But I want to focus really on antibiotic interactions. And I want to essentially set the stage for some things that Sada will talk about. But before I kind of jump into into the details of antibiotic interactions, I just want to show you, because I know that not everybody here, I think everyone here is interested in evolutionary medicine. Not everyone here really focuses on antibiotics and drug resistance. I want to show you how some drugs that have previously been considered to be not effective can actually be made effective in combination. Okay, and this example here is of an antibiotic that we have called vancomycin, and you might have heard of it. Sometimes the media calls it the drug of last resort. Of course, the media now calls a lot of things, I think, the drug of last resort. But vancomycin has been, I think, one of the original ones. This is a large glycopeptide, and it's usually well, it is not used in gram-negative bacteria because it cannot get through the outer membrane, right? So vancomycin is considered an antibiotic for gram-positive bacteria because gram-negative bacteria has this outer membrane that this very large glycopeptide cannot get through. And what I'm showing you here on the x-axis is different treatments. And this is just media with no antibiotics. This is with some amount of vancomycin. This is with some amount of a different antibiotic called trimethoprim. On the y-axis here is the percentage growth that the bacterial population does. And so in no drugs, it's growing at 100%. In some amount of vancomycin, you can see it's growing at essentially 100%. And the same thing with trimethoprim with a little bit more error, right? This is essentially showing that vancomycin by itself doesn't have an effect on a gram-negative bacteria. This is an E. coli. You can also see here that, oops, sorry, let me just, how do I make this smaller? Okay, let me just move this. That when you have this in combination, whoops, sorry, a little bit, you can actually see here. Can you guys see this? That that there is, you put these two together and suddenly there's this enormous effect.
Mm-hmm.
Okay.
Yes.
Okay. And so these two in combination can actually be extremely effective. And this is just a quick show to show why we're interested in combinations and how we can think about which combinations to use. And of course, this here you can see that's it's what we call a synergistic combination. They work very, very well together. These two drugs, vancomycin and trimethoprim. Okay. And so I just want to introduce how we can think about drug-drug interactions, how we can classify them. And there's essentially three types of interactions. One is no interaction or additivity. And here on the x-axis is time. On the y-axis is the density or the number of cells. And in the dark black line here, this is no drugs at all, right? This is like wild type with no drugs at all. And the blue line is the growth rate of the bacteria population with a small amount of ciprofloxacin, a type of antibiotic, one type of antibiotic. The blue line is the growth rate of the population with a small amount of erythromycin. And this is the growth rate of the bacteria population with the two drugs together. And here you can kind of just essentially see the growth rate there. And this is where there's no effect of one on another, right? It's additive because you can essentially add the effects of one on the other, and you'll get some growth rate here. What I previously showed you with vancomycin and trimethoprim is an example of an extremely synergistic combination. And this is another drug combination. And what you can see here, this is the no drug growth, the growth of just one drug, the growth in another drug environment. And then when you put it together, you see this dotted line and the population has essentially been killed off, right? And so this is an example of synergy. And then we have this very interesting example where two drugs work less well than you would expect given their single drug effects. And what you have here, again, is no drug growth. This is the growth of the bacteria in just one drug. And then you can see here, there's a blue line, growth of the bacteria in just one drug, tetracycline, but there's almost a complete overlap of the dotted line, which is also the two drugs together. And that's essentially saying whether you put in piperacillin or not, there's no effect of that. And so this would be an antagonistic drug combination. And so our lab is really interested in everything about these interactions. How do these interactions affect evolution? How do these interactions affect the likelihood of spontaneous mutations arising? And what happens further down the line? When we think about drugs, and drug combinations, the easiest thing for us to do, and which we focus a lot on, is just 2 drugs. But of course, we also know that a bacterial population often is exposed to a lot of drugs at once or a lot of stressors at once. And so what we've been focusing a lot in our lab the last few years is this question: what happens to a bacterial population that has a lot of stressors? And we consider these high order drug combinations. And so then the question really is, well, first of all, let me just think about what are high order drug combinations. The first is that anything with more than 2 drugs, or if we think about higher order stressors, anything with more than 2 stressors. We think that there's some potential clinical relevance, but my lab is not does not know very much about clinical work, and so this isn't really our strength. But what we do know is that it really speaks to a larger fundamental biological question, which is this: is the whole just the sum of its parts, or is it more, or is it less? And how can we make predictions based on some of these lower order interactions to understand what's going on at the higher order interactions? and that essentially is, let me just stop sharing. That's essentially what our lab works on. And what Dr. Boyd is going to talk about now is talking about when we add copper to the mix, when we add something else that's affecting the population, and what are all the changes that we might see, and how might it affect. I don't know that she'll talk too much about those interactions with copper and antibiotics, but she's going to talk about some of the genetics, and then some of the maybe there's some phenotypic changes and some effects. So I will turn it over to Dr. Boyd, who I'm so impressed by her work. And just another plug for those of you who weren't here at the beginning. She is on the job market this fall, and I'm very excited by what she can bring to any department. So.
Thank you very much. So thanks to all of you all that are involved in organizing. And also I want to thank my mentor. Pam is really wonderful, and I'm really thankful for all of her support. Can you all hear me okay? Okay, I'm going to go ahead. Okay, thank you. So I'm going to go ahead and share my screen. Okay, so today I am going to, the title of my talk is examining the evolution and interaction of antibiotic resistance and copper resistance in bacteria. And so here I am highlighting my funding sources. So I was funded by UCLA Erecta, and also I received the Burroughs Wellcome Postdoctoral Diversity Enrichment Program Award and the UC President's Postdoctoral Fellowship Program Award. And here, since I am on the job market, I have my contact information if anyone would like to contact me. So for today's talk, I'm first going to start with talking about the evolution of copper resistance, and then I will move on to correlated responses associated with copper resistance. And then finally, I will talk about the effect of copper and copper-dependent compounds on meropenem-resistant E. coli. So the motivation, or the reason I'm interested in studying copper resistance, is because over about the last 10 years or so, copper has been in the news a lot. especially being marketed as an antimicrobial surface. So there's been studies that have found that copper surfaces in fitness centers reduce bacteria and also possible infection. There's also news talking about how great of an antimicrobial copper is and how fast it works. Copper is also being implemented in hygiene-sensitive areas such as hospitals and airports, especially in healthcare, as far as these hospitals and clinics and nursing homes. And there's just a lot of information about copper and how it kills bacteria and how these copper surfaces are just really great at killing these superbugs. But there's also has been news about copper and how copper resistance have been in the making for thousands of years. And we're also starting to see that bacteria are able to survive on these deadly copper surfaces. So this is a reason that I'm interested in studying the evolution of copper resistance. So it's no doubt that copper can be used as an effective antimicrobial, and it can be implemented in these hygiene-sensitive areas to prevent the spread of bacterial infections. But we also need to think about that more exposure may lead to the emergence and spread of resistant microorganisms, similar to what we see with this antibiotic-resistant problem. So this brings me to my major research questions. First, what are the genetic changes that occur in response to the evolution of copper resistance? And does copper adaptation lead to resistance or increased susceptibility in the presence of antibiotics? And so the second question is really important when we think about our current antibiotic-resistant problem. And if we add copper to the mix, how is this going to affect this current antibiotic-resistant problem? So here is my experimental design to address these questions. So first, I just took E. coli K12 that is used for this study. I plated it onto standard Davis minimum broth, so it's a glucose-limited broth. I picked individual colonies, and these colonies founded populations that were separated into two groups. We had the replicate treatment populations, which were grown in only standard DMB media, and the replicate treatment populations, which were grown in a standard DMB media and a sub-inhibitory concentration of copper. So controls only DMB, treatment populations, copper plus DMB. And both of these populations were subcultured for about 37 days. And after 37 days, I took both of these populations, exposed them to varying concentrations of copper for 24 hours to determine if these replicate treatment populations now have some level of copper resistance in comparison to the control. And then I took these populations and sequenced their genomes and performed bioinformatic analysis to determine what genes are involved. So first, we found that these copper-selected bacteria have superior growth in comparison to the control and the ancestor. Although this is modest growth, they do grow better than the controls and the ancestors. So our copper-selected are these guys in blue. Our controls are these guys in red, and our ancestors. So the ancestors are just the populations that were used to found these controls and copper-selected populations, and these are the ones in green. And they were all exposed to these varying concentrations of copper. And so we see that these copper-selected bacteria, although they have some low-level resistance to copper, especially at these lower concentrations of copper. So now that we have established that these copper-selected bacteria do have at least some low levels of resistance to copper, I was interested to see what mutations may be associated with copper resistance. And so I sequenced the genomes of the controls and the ancestors and the copper-selected, but I only focused on the genes that were found in the copper-selected bacteria that were not found in the controls or the ancestors. So we found a bunch of mutations in the CPXA genes, CPXR, and CPXP. So CPXA is a sensory histidine kinase 2-component response regulatory system with CPXR. And these mutations that we found in CPXA were selective sweeps. So this means that 0% of the population went from not having these mutations to having these mutations. We also found selective sweeps in CPXR, which is this response regulator, and a few mutations in CPXP, which is an inhibitor of the CPX response. But all of the mutations in CPXP resulted in stop codons. So these mutations probably didn't actually take an effect on copper resistance. A few mutations were also found in Rho. But the mutations that were the most interesting to me were these mutations that were found in CPXA and CPXR. So just in case you're not familiar with CPXA, CPXA responds to envelope stress. So it receives this information about the status of the cell envelope. The CPXA autophosphorylates and then transfers its phospho group to CPXR. And then it induces a gene cluster responsible for protein secretion, cell envelope biogenesis, and quality control in the envelope. And so finding mutations in CPXA is actually was interesting because we're not the only ones that have found these mutations to be associated with copper toxicity. So finding mutations in CPXA related to copper resistance is not surprising, but it was actually pretty interesting. So after doing the whole genome sequencing and figuring out what mutations were associated with copper resistance, I wanted to know what are the correlated responses associated with copper resistance, especially when we think about antibiotics. So I took these populations, so these copper-selected bacteria that are in blue, these control populations that are in red, and these ancestors that are in green, and put them in varying concentrations of antibiotics related to protein synthesis, cell wall synthesis, and folate synthesis. And what we found is that the copper-resistant bacteria had increased sensitivity when compared to these ancestors and these control populations. So this means that there is some evolutionary trade-off in being resistant to copper and being in the presence of these antibiotics. And so this is important when we think about possibly using copper to treat these bacterial infections that are antibiotic resistant. So in summary, for this first half of my talk, copper resistance does occur rapidly. So it took about 37 days via de novo mutations. These mutations that were found were in the CPXA, which is important for envelope maintenance. And also that E. coli can rapidly evolve this resistance to ionic copper, and that this resistance may decrease the ability to grow in the presence of certain antibiotics. And so I do want to just say that I have started to redo this experiment in Pam's lab, and actually making this into a long-term evolution experiment. So I still have these strains to continue to expose them to copper to see what mutations may happen as we increase the amount of generations that are exposed to copper. So that was the first half, focused on the evolution of copper resistance and these correlated responses associated with copper resistance. But I'm also interested in these strategies to combat antimicrobial resistance, especially in the areas of drug repurposing and combination therapies, like Pam mentioned earlier. So in one area that I'm interested in is evaluating the effects of copper and copper-dependent compound on meropenem-resistant bacteria. So similar to the first experiment, I evolved E. coli to be resistant to meropenem, and grew these bacteria in LB in a subinhibitory concentration of meropenem for a certain number of generations until they were at least 4 times more resistant to meropenem in comparison to their ancestors and to the controls, and then performed 24-hour growth assays. So here I'm showing that these meropenem-resistant populations evolved to be up to 10 times more resistant than the ancestor. So the ancestral MIC of these two meropenem was about 0.125 micrograms per milliliter. But after evolution, or being constantly exposed to this meropenem, these populations evolved to be resistant of up to 10 to 23 micrograms per milliliter of meropenem. And so the next step was to take these meropenem-resistant bacteria and expose them to the single chemicals of meropenem, disulfiram. So disulfiram is actually an anti-abuse drug that is being repurposed as an antibacterial. And also I exposed them to copper only. And I should say that these are all sub-inhibitory concentrations that these populations were exposed to. The next step was to do 24-hour growth experiments using pairwise combinations. So taking disulfiram plus copper, so this copper-dependent compound plus copper, copper plus meropenem, and disulfiram plus meropenem, and then a triple combination of using disulfiram, meropenem, and copper. And so I wanted to see what is the effect of these different combinations on meropenem-resistant bacteria. And so when doing the 24-hour growth experiments in these CU compounds, copper only, copper plus disulfiram, and disulfiram didn't have any effect on these meropenem-resistant bacteria, which you can see here. But if you expose these populations to copper plus meropenem plus the copper-dependent compound disulfiram, and copper, meropenem, and disulfiram, we see that it has an effect. It affects the relative growth of these meropenem-resistant bacteria, and it affects them at the level in which the meropenem is not normally able to affect the ancestor. And so if you remember, the ancestral MIC was around 0.125, but the amount of meropenem that they were exposed to in these combinations, or that were able to kill them, were around 0.08. And so this is showing that in the presence of copper and disulfiram, meropenem's susceptibility increased for these meropenem-resistant bacteria. So in summary, the preliminary results suggest that synergism among copper plus meropenem, meropenem plus copper, and the triple combinations of copper, disulfiram, and meropenem. I am currently still working with these strands and doing further studies that will include measuring the interactions and also determining the potential targets of these combinations and the use of more clinically relevant strands such as Pseudomonas aeruginosa. So so here I do have my acknowledgements. So I first want to acknowledge the Graves Research Group, which is the start, which is where I started these evolution of copper experiments. Also want to acknowledge Dr. Pamela Yeh, where I did my postdoc at UCLA, and a few of my collaborators and undergraduate researchers. So I went actually only about 15 minutes, so I'm very open to any questions, and if any questions about future projects and the research that I presented just now.
Thanks a lot, Sada and Pamela. So the floor is open for questions. To get everything going, perhaps. I find it very interesting that you found the CPX system to be important here in copper resistance, and we actually have been working on the CPX system in a slightly different context. So I was wondering, first of all, is it gain of function mutations you see in CPXA, or is it loss of function? And do you have any information how relevant the inner membrane stress is itself, or is it some other kind of signal that is perceived and relevant in this context?
Yeah, so we I suspect that it is a gain of function mutation for CPX, because most of these mutations were found in the hemp domain. So I was like, what I think is happening is that the CPX mutations are allowing CPX to be constitutively activated, so that it can respond to the stress of the copper. And I'm sorry, your other question was about inner membrane.
Yeah, it was inner membrane stress, whether, I mean, that's not really trivial how to measure that, but if you have any indication whether it's inner membrane stress that is relevant here as a signal, or whether it may be some kind of other signal that is perceived here.
So yeah, so I'm not sure, but I think it's just the stress of copper actually attacking the outer membrane, which is causing this activation of the CPX. And then once there's, of course, when there's too much copper in the inner membrane, it starts to bind to these other components of the bacterial cells. So I think it's initially the outer membrane stress, because copper does target the cell envelope.
Okay, thanks a lot, Shade. Christina has a question.
Yes, thanks. Thanks a lot for the talk. So I'm from the agricultural side, and we have been using, not us. Can you understand me?
Yes, I can.
Okay. So they have been using copper a lot as a substitute for growth promoters after they were prohibited, and we saw cross resistance rising, so ampicillin, some other penicillins. So did you also do research on that side of the matter?
As far as the cross resistance?
Mm-hmm.
Yes, so actually that is what I didn't present that today, but I have done some cross resistance and susceptibility tests with a range of different antibiotics. So it does seem to be that it is mechanism of action dependent. So depending on the antibiotic, whether it is more susceptible or resistant to this antibiotic or copper.
Okay.
So did that answer your question, or I'm sorry, I want to make sure I understood you.
Yeah, no, it's fine. I'm just, so all colleagues are wondering where that comes from, that cross resistance. So they're doing research. Is it mutation based? Is it on common mobile genetic elements? And it's not that clear. So I thought maybe, but your research is mostly on the mutation and evolution point. I think so.
Yes. So actually, so we see both. We see that it can be because of mutations that are on the chromosome, but also horizontal transfer of these genes. And then also some of it is just because there is the same mechanism of action. So the same, it's more of like this generalized stress response of between the antibiotic and the copper. So we see it in different ways, but for the project that I just presented, we were only focused on seeing the mutations on the chromosome and haven't paid too much attention to horizontal transfer. But there are other studies that have shown that these genes can be horizontally transferred.
Okay. Thanks a lot. Johnny, you have a question?
Yeah, thanks, Shade and Pam. Kind of just a comparative, sort of relative question with other antimicrobial metals. I think in the health world, you think of zinc, and I don't know if that's like paired with bacitration or anything, but like zinc and silver, how does that, like the literature, align with what you're finding? And also, what do you anticipate given, like, I think zinc and silver probably, and copper are the three most prominent, but I don't know if there's others out there. I'm just wondering, does this kind of align with your expectations, or anything that was kind of striking?
So I think for like your first part, I think it does align with what people are saying with silver and with zinc. So I know at least for Dr. Graves' group in working with silver resistance, they also have seen some cross resistance and some increased susceptibility. But I do think that the results are not extremely surprising. So I did expect to see some of the mutations that we saw, but also I was expecting to see other mutations. So mutations in like CUS, which are copper sensing proteins, which we didn't find. But also I have repeated this experiment in Pam's lab, and I actually didn't find any mutations in CPX. And all of these other mutations were associated with like mechanosensitive channels, which is an avenue that I'm trying to explore now.
Awesome, thanks.
Did that answer your question?
Oh yeah.
Okay, thanks. Are there any more questions from the audience? Then I will come up with another point, because what I find really intriguing is your that you start now combining 3 drugs. It is interesting in a way because this has been neglected in the basic research field and in the evolutionary research field, and I think for good reason, because combining 2 drugs, there we have actually nice theoretical frameworks. We have clear concepts how to measure synergism, antagonism. There's the concept based on Bliss independence. We have Löwe additivity. We have the Ficki index and so on. So how can we actually conceptualize the interaction between 3 drugs or even more? I mean, one standard tool also is that we produce checkerboards with increasing concentrations of 2 drugs. So when you then add a 3rd or 4th drug, you need a 3-dimensional checkerboard or a 4-dimensional checkerboard. How do we do that? Do we have to rethink these concepts? Pamela, Sada?
Yeah, so this is a great question. I agree. Like I think the reason it's taken the field of evolutionary biology so long is the difficulty in figuring out the null. What is the null model?
Yes.
And how we've been doing it is thinking about subtracting all of the lower order interaction terms. And so, for example, for a 3-drug combination, we would first subtract out the single drug effects, and then we would subtract out the 2-drug, all the pairwise combinations. So in 3 drugs, there would be 3 pairwise interactions, like drugs A and B, drugs B and C, and drugs C and A. And then we see what's left, and we call that the emergent interaction. So the net interaction is just overall based on the single drug effects. Is it more or is it less? But that's not, and I think that's important, but that's overall not as interesting, because I think that the net interaction could be swamped out by a very strong 2-drug interaction, or a very strong single drug drug response. And so if you can subtract out this all the lower order interactions, then you can see what's left, and then you can say, is that a synergistic combination, or is that an antagonistic combination? Or if there's nothing there, then it's we term that additivity, and that has been for us, it's enabled us to say, well, this is a real interaction that requires all 3 drugs. It's not, it might look antagonistic, and its net interaction might be antagonistic, but that might be because of 2 drugs, or it might be really synergistic, but that might be because of these 2 drugs. And so if we can subtract out all of the lower order interactions, then we can say, actually, this is a result of these 3 drugs interacting. And that gives us the framework for being able to then, just like people have done for 2 drugs, to start to look at a set of 3 drugs and say, is that something that's relevant? And in part because particularly in, I think in the evolutionary biology world with looking at stressors, people have always said, and I don't know if this is as true for the drug world, but I think people have always said, thinking about stressors, well, the higher order ones are too difficult to examine, particularly in the field, and they're probably not very important because they're probably all canceling each other out. And on top of that, they're not important probably because they're being swamped by the major interactions between two stressors. And so this is, for us, we've been able to look at using this new framework. We've also then been able to take it into the non-drug world and think about the stressors there. I'm sure there's more that can be done with this, but that's one way that we've been able to think about looking at the looking at how we can imagine higher order interactions.
So when you experimentally explore these interactions, do you then perform three-dimensional checkerboards, or you do? Okay.
We do, yeah, and it's very messy. We and we use robotics, and I think I believe we did it for about 8 drugs, and all the 3, 4, and 5 drug combinations, just to ask. Well, we first looked at 3 drugs, and then we looked at 4 and 5 drugs, and all we wanted to do was ask a very simple question: are there more or fewer or no changes in interactions as you increase the number of drug combinations? Because, there's this idea that as you increase the number of combinations, you don't necessarily increase interactions because maybe it's all additive. They're so small. They're all, and so we just wanted to see that. And what we found is that there are, it does increase. There are both synergy and antagonism. It's not that they're all additive. So that was the first thing that we found. And the second thing that we found is that there's an increase in antagonism in this as far as an emergent interaction. And so then you want to be really careful from the clinical side as you add more drugs that there becomes these emergent antagonistic interactions if what you want are synergistic combinations. So, and then there are some people who are really interested in antagonistic combinations, and that's another way to have more antagonistic interactions is to add increase the number of components.
Thanks a lot. I mean, I have follow-up questions, but I still want to check whether there are more questions coming from the audience. Then I will come up with my follow-up questions. So the other interesting point is, of course, in the end, we do our kind of basic research to, in the end, be able to transfer to medical treatment. What is your overview at the moment? How much are sort of combinations of more than 2 drugs used? Excluding TB, so there it is very well established. Excluding HIV, it's very well established. But for infections with E. coli or Pseudomonas, other of the escape pathogens, do actually, in clinical treatment, for example, in the US, do people actually do a combination of more than 2 drugs?
That is such a great question. And I saw Mark Iravani here, but I don't see him anymore. Maybe he had to leave. But he is much more on the clinical side, and there are other folks. So I try to be very careful talking about the clinical side because I know so little about it, and I'm worried I'm going to say something wrong. But I'm gonna do my best, which is my understanding is that there isn't a lot, but that in general the clinicians understand that as we get into more drug resistance and mutants that are extremely resistant to the drugs that we have, that this is the direction it's going to go. It's not just a single drug. It's not just 2 drugs. It's going to be more drugs. I have a personal interest in antagonistic drug combinations because I think that for just a simple fact that they give very strange and unexpected fitness landscapes. And so I'm really interested in how that might be useful in the clinical side. From what I've been told, I haven't talked to a ton of people, but I've talked to enough. They're very disturbed by the idea, and they think it's not a great idea. But there are some people who are maybe interested, but for the most part, the general reaction is we don't want to do antagonistic drug combinations. So we want to be very careful that we don't want to actually use it. But I think that there are some labs that have become really interested in these higher order combinations from the clinical side. And I think that's in the minority. So I don't know very well, but I'm really curious outside of, Los Angeles and California, if that's something that other clinicians and hospitals have been thinking about, if anybody knows.
Yeah. So I don't know if it's actually being used in the clinic yet. I know there's like interest in using it. And also at the end of my presentation, I was talking about using these more clinically relevant strains. So I am going to be doing this with a clinically isolated Pseudomonas strain of using these antibiotic plus these copper-dependent compounds. That's still very preliminary, so I didn't want to try to to present that today, but is it interest? I don't know if it's actually been translated to be used clinically yet.
So the comment that I get from clinicians when I raise that point is that they're afraid of using more than 2 drugs, because with the argument that if a bug survives and evolves, then you immediately have a super bug. And that is, of course, an interesting point, but you could still argue you could still leave some kind of drug behind. So let's say you don't take a peptide drug, but only combine other kinds of drugs. And that may be more efficient, as the example of TB shows, where this is very well established. I also find it curious that this response, because when we now talk about other strategies to fight pathogenic infections like phage therapy, there it is usually the argument that we should use cocktails of different phages. And with that same kind of reasoning, one should also think of using cocktails of different antibiotics or other drugs.
Yeah.
Okay, I have a look into the audience. There are no more questions. And I think we can close the session for today. So I very much would like to thank again Sada and Pam for a very inspiring presentation. And yeah, I hope you'll join in next time again. And also do not forget activities of our ISEMP Society of Evolutionary Medicine and Public Health. I should actually remind you there is this year's meeting will take place in Durham in the UK from 6th until 9th of August. And it's a fantastic opportunity to exchange, get into contact, catch up with each other. So please join us there. And with that, I thank you all for being here today.
Thank you.
Thank
you.
you.
Thank you.